Chapter 24, last chapter of the semester, is going to be on genetics and inheritance and some diseases and so forth. And we start out here with a couple of definitions. Genetics is defined as the study of inherited characteristics, or the process of inheriting different characteristics, okay? Genes are DNA sequences, okay? So sequences of DNA that encode for proteins, okay? I want you guys to get big picture information right here. The purpose of genes, the purpose of your DNA is to produce proteins, okay? Proteins are going to be what actually causes your traits, okay, or causes what we call your phenotypes, and we'll get into that a little bit more. But it's the DNA that codes for proteins, and the proteins is what makes you have a widow's peak or not, or blue eyes versus brown eyes, or I don't know, connected versus not connected earlobes, and so forth. Another term here is genome, and a genome is a complete set of genetics and genetic instructions, and it says in a person's cells. And one thing that I think is kind of neat is that all of your cells, whether if they have a nucleus, right? Remember, red blood cells don't have a nucleus, but all cells that have a nucleus have every single set of genetic instructions. So for example, the cells in your eyelashes that make up your eyelash follicles will have instructions for producing saliva. Okay, I think that's just really kind of interesting. But those instructions are not read and used to make proteins. Obviously, your eyelashes don't make saliva. Okay. A genome is written in DNA language, which is, if you've had micro or general biology or biology in high school, you remember A's, T's, G's, and C's, adenine, guanine, cytosine, and thymine, okay? The order, the sequence of these nucleotide bases is what makes up your genome, okay? What makes your genes different from my genes is their sequence. Okay, and another interesting thing is that human genomes, okay, whether yours or mine or someone of a different race, someone of totally different background, all of our DNA is 99.9% the same. The thing that makes us different is how those genes are expressed, or how those genes are made into proteins. Okay, how and when they're made into proteins, and also that other 0.1 or less %. Okay, another example of a term here is exome. So we have genome, which is the complete set. The exome is only the protein coding genes. Okay, and that's about 2% or 20,000 genes. Okay, less than 2% of our genome. I'm kind of running out of room here. The rest, 98%, are genes that control the exome. Okay, the other 98% are the genes that control when and how those 2% of the genes, the exome, are going to be made into proteins. Let's see. So this process of making genes into proteins is called gene expression. Okay, where you take DNA, turn it into RNA, and then RNA is going to produce the proteins. Okay, and so that is called gene expression, where we have DNA is transcribed, if you remember that term, into RNA, and then RNA is translated into protein. And those proteins then produce the trait or the phenotype. Okay, now our traits are not only for the most part they're produced based on our genetic information, but the environment also can affect how and when those genes are expressed. Okay, so remember, anytime I say gene expression or genes are expressed, that means we're going through that process of taking the genetic information and turning it into a protein, which can then cause a trait. Okay, so here is a diagram, and I like to really show this one. This is a dividing cell. Okay, now a cell that's going through mitosis or meiosis. And during that time, you can see the little X-like chromosomes that we typically think of chromosomes. When the cell is not dividing, okay, most of the time, this DNA is kind of in a squiggly mess, and you cannot see individual chromosomes. So what we're looking at here, you can see the little X-shaped structure here. That DNA is all condensed down into that nice little X, and that's the chromosome. And the chromosome is basically wound up. This is all DNA here. The chromosome is DNA wound around proteins. That's those purple structures here. There's I think there's like 8. They're called histone proteins that they wind up and wind around. So you can see the DNA, how it's wound around these little proteins, and that's what makes up the chromosome. Now, a gene is a segment of DNA within the chromosome. So this is, say, gene number 1. Okay, here you have gene number 2. Okay, and you'll have dozens or hundreds of genes on, or thousands even maybe, on a single chromosome. Okay, so a region of the DNA is going to be a gene. Okay, and you're going to have A, T, G, C, A, these sequences of those nucleotide bases. Okay, oh, I wanted to make one other point here. I don't know, we have enough room all over. Get that all erased there. Remember the gametes, or the sex cells, the egg and the sperm? The gametes are going to have one copy of each chromosome. So they're going to have 20, in a human, 23 chromosomes. In our non-gamete cells, those are called somatic cells, you have 2 copies. So you would have 46 chromosomes. Okay, so maybe some of you guys back in high school or general biology course learned about Gregor Mendel. He was an Austrian monk that studied pea plants, and he discovered that genetics can be predictable. You can do certain crosses, and you can do some math and figure out the probability of parents with specific traits, the probability of what trait their offspring may have. And so there are patterns of inheritance, and that's one of the major things that Mendel studied. And these patterns in which genes are transmitted in human families is called modes of inheritance. Okay, and we'll talk a little bit about some of the probabilities and go through some Punnett squares and things here in a little bit. But a karyotype is basically this picture, or it's defined here as a chart of chromosome pairs based on their size order. Okay, so our chromosomes are numbered based on their lengths. Okay, so here is a normal karyotype, or a normal picture of our 23 chromosome pairs. Okay, now the first 22 are called autosomes. Okay, these are the non-sex chromosomes. Okay, the 23rd pair is the sex chromosomes, and that's going to determine gender. Okay, and we consider XX female and XY to be male. And I can tell that this is a female because she has 2 chromosomes here of the same length. Okay, and so that we would represent her gender as XX. Okay, if it were a male, you would have a long chromosome for the X, and then a shorter chromosome, which is represented by the letter Y. Okay, so humans have 23 pairs of chromosomes for a total of 46. Now, if we were looking at an egg cell, or an oocyte, or a sperm cell, that cell would have only one of each of these. Oops. Okay. Now, when the egg and the sperm join together, then we have what we call a diploid, or a 2-copied zygote. Okay, so we'll pretend this is a zygote right here. And this on my right here is going to be from dad, and this one is from mom. Okay, and then this one would also be from dad, and this one would be from mom, and so forth. Okay, so this is what your karyotype would look like. Okay, 1 of number 5 came from your mom, 1 of number 5 came from your dad, and 1 from 9 mom, 1 from 9 would also have come from dad. Okay, and you can see these little bands here. Okay, this is a gene that controls something. Here's a gene for something else. There's all kinds of genes on each one of these chromosomes that are used to either control other genes or code for proteins. Okay, and so all of these chromosomes here, right here, contain all of the genetic information needed to create you and to give you your unique characteristics. Okay, so that chart is called a karyotype. Chromosomes 1 through 22 are autosomes, and then the 23rd pair is the sex chromosomes. Okay, so as I mentioned before, each chromosome has hundreds or thousands of genes on it, which are, again, regions of DNA. And we said each human cell, other than the gametes, have 2 copies of each chromosome. Again, 1 from mom, 1 from dad. Okay, and each member of the pair, this word homologous chromosome, so this one and this one, each of those are called homologous chromosomes. This one and this one is a homolog. This one and this one is a homolog. Within each type of gene, okay, or within each gene, there's some variations of that gene, okay? Because remember, we don't all have brown eyes. We don't all have widow's peak. Some people do, some people don't. And so there's variations in genes out there. And those variations or alternative forms of a gene are called alleles. And alleles are going to differ slightly in DNA sequence. So, for example, for widow's peak, you might have TAGC, blah, blah, blah. Okay? That's the DNA sequence that makes up the gene that causes widow's peak. A person without widow's peak might have A, let's see, C. And then everything else is the same. Okay, so it's just slightly different in its sequence. Okay, so this one's going to cause a straighter hairline. This one's going to cause a widow's peak hairline. Okay, so with the alleles, if a person has two identical alleles, they're called homozygous. So homo means the same, hetero means different. Okay, so let's use the widow's peak for an example. So if a person is, I think widow's peak is recessive. Should have looked that up. We'll pretend it's recessive. Okay, so say a person is, we're going to represent their DNA sequences by letters. Okay, so say a person has two normal hairline, straighter hairline alleles. Okay, they would be homozygous. If they were the same here, where you had two widow's peak alleles, you got this one from mom, this one from dad, that would also be homozygous. This one would have a normal hairline, this one would not. A person that's heterozygous would look like this. They'd have a normal one and a widow's peak allele. Maybe they got this one from mom and that one from dad, or vice versa. Okay, so homozygous is having two of these same identical alleles. Heterozygous is having one of each of the alleles, or two different alleles. Okay, so your combination of alleles determines what is your genotype, or your genetic makeup. For a trait. Okay, so your genotype would be big N, little w, for example, or big N, big N. What you look like, okay, the appearance of that trait is your phenotype. So you would, phenotype in this example would be widow's peak or straight hairline. Okay, so the appearance is the phenotype, the genetic makeup is going to be the genotype. Okay, then we have what are called wild type alleles, which are the most normal or most common in the population. And then there's mutant, and that's basically an allele that's very uncommon, and it may or may not cause a disease. So like we'll talk about here in a little bit, cystic fibrosis. The cystic fibrosis allele is not very common, luckily, and it would be called a mutant allele. Okay, so as you've probably learned in general biology before, in high school or wherever, you've probably learned about dominant and recessive characteristics, or dominant and recessive alleles or traits. Okay, so remember, if a person is a heterozygote, they're going to have two different alleles. Okay, uppercase is usually referring to dominant, lowercase refers to recessive. So a lot of times, if you have two alleles that come together and are going to mask another trait, that's called a dominant trait. Okay, so I used a big B, little b here. Let's pretend we're talking about black and chocolate labs, dogs. Okay, big B is going to represent black fur color, little b represents brown. Sorry, this is brown. black hair color is dominant. So anytime there's a black allele, it's going to mask the little b. It's going to mask brown. So if there's ever a dominant allele, it's going to overshadow this, and that dog is going to appear black. It's going to have a black phenotype. Okay, you only need one allele. Okay, so if you have this, that individual is going to be black, or this is going to be black as well. Okay, because of the dominant allele. Now with a recessive allele, you have to have both recessive alleles present to get the brown color. So a brown phenotype or a recessive phenotype can only look like this. You have to have two recessive alleles. Okay, now diseases can be caused, genetic diseases can be caused by either dominant or recessive alleles. Okay, and we'll talk about some of those here in just a little bit. Okay, so again, that term autosomal, an autosomal allele is going to be carried on a non-sex chromosome, which is chromosomes 1 through 22. If we have an X-linked allele, that's going to be carried on the gamete or the sex chromosome. On the X chromosome, and a Y-linked allele will be on that 23rd chromosome on the Y if it is a male. Okay, now autosomal conditions affect both genders equally. Okay, but if you have an X-linked or a Y-linked disorder, that's when males or females, depending on the disorder, may have a higher risk than the other. Okay, so a person that carries a disorder, an allele for a disorder, and that condition is recessive. Okay, so for example, if we look at cystic fibrosis or CF, okay, that is a recessive disorder. So in order for a person to have a recessive disorder, you have to have both the CF alleles. Okay, now a person that is normal, has a normal allele and this CF recessive allele, remember this dominant allele will mask this and they will appear normal. Okay, so if someone is married or has children that are heterozygotes, okay, we have my thing's not erasing, that's weird. We have a heterozygote, so both of these parents are going to be normal. Neither one of them have cystic fibrosis. They're what we call carriers because they carry this allele. And if these two alleles happen to cross in the offspring, they could have an affected child. Okay, and so two heterozygous parents that have a recessive allele in a recessive disorder like cystic fibrosis, those are called carriers. So if you have two carriers, there is a chance that a child could have the disorder. And these conditions can skip generations. Okay, so maybe grandma or grandpa had the disorder, children didn't have the disorder, but then grandchildren could if the right combinations of alleles line up. Okay, really not sure why we're not erasing there. Anyway, with a dominant condition, okay, you only have to have one affected parent. You only have to have one of those affected alleles, and these do not skip generations. Okay, and we're going to go through some examples here in just a second. But we have some tools that can predict the probability of two parents having offspring with a particular type of a trait, okay? And so back in the day before genetic testing, Punnett squares and pedigrees were very, very valuable tools, and they still can be today, but we have much more sophisticated technologies, and we'll talk briefly about those at the end. But a Punnett square, and I'm sure you've all done these before, a Punnett square is going to predict future offspring, okay, or their chances, okay, used to predict future probabilities of genotypes for an offspring, okay? A pedigree is going to look back at the family history. What did great-great-grandma have? Was she a cystic fibrosis person that's afflicted? Was she a carrier? You know, did she show signs of the disease? Okay. So a Punnett square, as you know, if we're looking at one trait, is basically a set of boxes, and we cross the traits, and we'll go through some examples here. And we look at all the possible combinations of alleles at fertilization. Okay, so each little square represents a possible zygote. Okay, we'll go through those here in just a second. A pedigree looks back at genotypes, if those are known, or phenotypes of a family member. Okay, circles represent females, squares represent males, shaded circles or squares represent someone that's affected by cystic fibrosis or hemophilia or whatever. Half shaded circles or squares makes a carrier, and then unshaded would be unaffected. And remember, carriers have the allele, but they're unaffected. In recessive disorders. Okay, so let's take a look at an example here. Cystic fibrosis is an autosomal recessive disorder. Okay, so that tells us the mutant allele, okay, the cystic fibrosis allele is located on an autosome 1 through 22 chromosome. Okay, so the wild type allele is the normal healthy allele, and they've got it represented by a plus here. Okay, so in this particular cross, they're looking at two heterozygous parents. And remember, for recessive disorders, heterozygous parents are carriers. Okay, these are unaffected by cystic fibrosis. They carry the allele, but they do not have the disorder, because remember, you have to have two of the alleles to get this recessive disorder. Okay, so we can look at the possible combinations of the alleles in their offspring. Okay, so if we took the egg from mom and the sperm from dad, okay, mom's eggs, one could have a normal wild type allele and the other could be a carrier, okay, could carry that cystic fibrosis allele. Same with dad. You could have this, or you could have that in a sperm cell. Okay, so if these two unite, that egg and that sperm were produced, you would get an unaffected non-carrier. Okay, if this and this one united, or if this one and this one united, you would get an unaffected carrier, okay? Because this child carries the allele, carries the normal allele, but they would not be affected. These children would not have cystic fibrosis. There is a 1 in 4 chance, or a 25% chance, that this allele and that allele would combine and the child could be an affected carrier, okay? or I'm sorry, an affected individual. They would have cystic fibrosis. Okay, so looking at here, okay, this individual is going to be a homozygous dominant, healthy, non-carrier. These two here are heterozygous dominant carriers, but they're not affected. And then this one would be a homozygous recessive affected person with cystic fibrosis. Okay, so I want you to take a look here. This one is homozygous because it has 2+ alleles. And this one is also homozygous because it has the 2 recessive alleles. So this has 2 recessive alleles, this one has 2 dominant alleles. And so that, because they have identical alleles, they're going to be homozygous. These have different alleles. They have a plus and a minus, or a plus and a CF. So those are 2 different alleles, so that makes them heterozygous. Okay. Another way that you can look at this is using a Punnett square. Okay. So if we have mom here on the top, she has a plus allele and a CF allele. Dad has a plus allele and a CF allele. Okay. So these are the possible gametes from mom and from dad. And if we look at those combinations, this is just another way to look at it in a Punnett square. If you put the 2 pluses in here, put a plus and a CF here, plus CF here, CF, CF. Okay, you just take a second and look at how these are combined, okay? Because you will have to do Punnett squares on exam 5, okay? So you take the top and the side and combine it. Top and the side and combine. Side, top, side, top. Okay, so then if you look at that, you look at all the combinations, you have one that's a homozygous dominant, one that's homozygous recessive, and then you have 2 that's a heterozygous dominant. Okay, so your chances are 1 out of 4, 2 out of 4, and 1 out of 4, or 25, 50, and 25%. Okay, so I want you to make sure you understand that. So take a few minutes and look that back over. Okay, now if we were to look at the a pedigree, okay, pedigree would be looking at heterozygous carrier, and then you would be looking back at the children that they had. So this would be later on, okay? Maybe these would be the grandparents or something like that, okay? So this isn't a prediction, this is looking back. And so Tina, we can know that since her shade, or her circle is shaded in, she's a female, since it's a circle and it's shaded, she had cystic fibrosis. Sue is half shaded, as well as Mary and Joe. Those half shadeds are carriers. And then Bill is going to be unaffected because there's no shading there. Okay, so this was for an autosomal recessive disorder. Okay, and just really quickly about cystic fibrosis, the allele for this disorder is found on chromosome number 7. And what cystic fibrosis is, like it's defined down here, it's a genetic disorder where there's a chloride ion channel in the cell membrane. This channel protein here that makes up that channel is defective. And so chloride ions are not transported across the cell into the cell correctly. And that results in thick mucus buildup in the lungs and the pancreas. And typically, maybe you've seen the movie 5 Feet Apart, or maybe you know someone with cystic fibrosis. They get a lot of mucus that builds up in their lungs, and that can really cause some problems with breathing. They have very, very sweaty salt, and there's other aspects of the human body that it affects as well, but the most significant would be the clogging of the airways because of that mucus buildup. And it's all because this gene codes for a defective protein that does not pump chloride ions properly. Okay, now another type of disorder is an autosomal dominant disorder. Okay, cystic fibrosis is recessive. This is a dominant disorder, and Huntington's disease is characterized by loss of coordination, behavioral changes, cognitive or mental ability declines, and a person with Huntington's disease may have uncontrollable dance-like movements. They cannot control their muscles. Okay, now since this is a dominant condition, a child only needs to have one affected parent, right? Because with a dominant condition, you only need one allele. With cystic fibrosis, you need two of them to get the disorder. With Huntington disease, you only need one allele to get the disorder, okay? So here we have an example, okay? Maybe put a star by that. You only need one mutant allele, okay? So we have an unaffected mom here, which she has the wild type alleles, okay? She has the normal, quote unquote, normal alleles. And dad has Huntington disease, okay? So he has a wild type allele, a normal allele, and one of the Huntington alleles, okay? So if we look, mom's egg, one would be plus, the other allele would have a chance would be a plus, okay? Dad's sperm, though, could be plus or Huntington's, okay? So here's what we would have in our Punnett square. Mom could be plus or plus. Dad could be plus or HD. So if we go top and side, you get plus plus or HD plus, HD plus. So you basically have a 50-50 chance of having an affected child, okay? Now the pedigree looks a little bit different here. Okay, affected person, non-affected person. Affected, non-affected. There aren't carriers when it comes to autosomal or dominant disorders, okay? You either have it or you don't, okay? Now, one thing is to think about too, or another telltale sign of a dominant disorder is that these symptoms appear in adulthood. Okay, so you may be thinking, well, if this is Dan, why did Dan have kids if he knew he had Huntington disease? Okay, well, maybe he just wanted to take the risks and they really wanted to have kids anyway. But there are a lot of times a person may not know that they have Huntington disease because they don't show up and don't have any symptoms until after they've already had children. Okay, and I'm going to back up one. With cystic fibrosis and other recessive disorders, these mostly appear in childhood. Okay, so recessive disorders a lot of times will appear in childhood. Those symptoms will develop. Dominant disorders will appear mostly in adulthood. Okay, now I have a blank slide here, and maybe you want to grab a blank piece of paper, an extra piece of paper to give yourself some room. I'm going to give you an example, and we're going to work through a Punnett square. Okay, so the first example here is we have in this example goes through hair color, and hair color is one that we'll see here in a little bit, like eye color and height, that is controlled by multiple different genes. or alleles. So, but we're going to look at this kind of on a basic level. Okay. So mom is going to have blonde hair, which is recessive. Okay. Dad has brown hair, and that's going to be dominant. Okay. So if dad is homozygous, okay, he would be big B, right? Brown is dominant, so he's homozygous here. For this trait, what are the possibilities for their children? Okay, so dad's hair color is brown. Oh, that's kind of confusing. So he will have alleles that look like this. That would be his genotype. Mom's hair is blonde, which is recessive. So we'll call her genotype little b, little b. Okay? Now, if we set up our Punnett square here, okay, we put mom's possible allele here and here, and then we put each of dad's possible alleles there and there. Now we can fill in our Punnett square. So big B, little b, here and here, big B, little b. Pull this one down, this one across, to down and across. We're going to get a child that's going to be, or there's a 100% chance that the child will be heterozygous. And have brown hair. Okay, because, excuse me, this child has one, or each possible zygote there is going to have one of the dominant alleles, which automatically gives them brown hair in this example, and each one of them is going to be a heterozygote. Okay, what if we take another example, and mom again is going to have blonde hair, so she's going to be little b, and dad is going to be a heterozygous. So dad looks like this. Okay, so we'll set up another Punnett square. Okay, so mom here again can be little b, dad can be big B or little b. Okay, now the most difficult part of this is figuring out what letters to put here on the sides and the top. Okay, and it doesn't matter if you put dad on top or mom on top, it doesn't really matter. But what you're going to look for is all the possible allele combinations for mom. Okay, so mom could only be little b or little b. Okay, dad has a big B and it has a little b. Okay, so then we take this big B across, little b down, big B, little b. Now we have little b, and little b. So in this situation, our chances of a zygote being brown hair versus blonde hair is a little bit different because dad is a heterozygote. Okay, so dad, a homozygote up here, all of their offspring will have brown hair and be heterozygous. Here, we're going to have a 50% chance, or 2 out of 4, that they're going to be a heterozygote, brown, and a 50% chance that they will be homozygous recessive, blonde. Okay, so on the test, I may give you a question where it says, okay, you know, this person, mom has this particular trait, she is heterozygous dominant. Dad has this other trait, he is heterozygous dominant. You know, figure out the probability of what their offspring will be, and you would use a Punnett square. Okay, if you have questions on this or want me to go over it differently or again with you, please reach out to me and let me know. And a little later, we're going to look at another example of a Punnett square. But these right here are pretty basic compared to what we do in some other genetics or biology classes. So I definitely want you to be able to put one of these together and figure out probabilities. Okay. All right. Now, a lot of times when we look at Gregor Mendel and his experiments on peas and so forth, and even this example that we just did on hair color, things may seem very cut and dry, okay? If it's dominant, it's gonna mask completely, and only one allele, or I'm sorry, a gene causing this trait. But in reality, not all traits are completely dominant. Not all traits are controlled by one gene. Okay. Sometimes we have genes, there's exceptions to Gregor Mendel's rules. Okay. We have sometimes what are called incomplete dominance. Okay. So in the cystic fibrosis and Huntington's disease example, there's complete dominance. Okay. So the dominant allele is completely dominant and completely masks the recessive allele, but not in all traits. Okay. Incomplete dominance is when you have a heterozygote. Okay, so HH is completely dominant, but you have a heterozygote. That individual is going to be intermediate between the dominant and recessive alleles. And an example that we use in general biology is if you take a white flower and you cross it with a red flower, and it's incomplete, the gene is incompletely dominant, then you would end up with a pink flower. Oops. So, okay, so it's kind of halfway or intermediate in between the dominant and the recessive individuals. An example in human anatomy is familial hypercholesterolemia, or familial genetic high cholesterol. Okay. So in a normal person, okay, normally if you go and you have your blood tested, they'll want you to have your cholesterol, your total cholesterol, below 200. Okay, and that's milligrams per deciliter. Okay, so 200 here is the recommended, for health, to prevent cardiovascular disease. You're going to want this number to be below. Okay, having high cholesterol can cause heart problems, cardiovascular disease. Okay, and familiar hyper means more. Familial hypercholesterolemia is basically a genetic disorder that gives you or causes you to have high cholesterol. You may have great exercise routines. You may have a low cholesterol diet. You may not eat bacon and eggs for every meal. Maybe you never eat that kind of thing. But no matter what you eat, your cholesterol is always going to be high. And what happens is, in a person with a quote unquote wild type, normal type of genetic profile, they will have an abundant number of receptors on their liver to grab cholesterol out of the bloodstream, pull it in, and metabolize it. Okay. A person that is a homozygous for this disorder, okay, homozygous for familial hypercholesterolemia, they have very, very few receptors on their liver to pull cholesterol out of the blood. Okay, so any cholesterol that's in the blood remains in the blood and is not pulled out like it would be in a normal functioning liver. So they don't have the receptors on those cells to grab onto the cholesterol and pull it out of the bloodstream. And so then they may get fat deposits around the eyes, at an extreme risk for cardiovascular disease, fat deposits in the joints, and lots of problems. Look at here. They may be up to 800, 900, or 1,000 milligrams per deciliter when you're supposed to be below 200. Okay. Now, where we get into that incomplete dominance here is that a heterozygote will have half of the normal number of receptors. Okay, so here's normal, here's none. These guys are halfway in between. Okay, and so a normal cholesterol for them might be 300 or 400. Okay, so the phenotype for a heterozygote is intermediate between the dominant and the recessive phenotypes. Okay, another exception to the dominance, the dominant recessive kind of rule is codominance. And codominance, instead of being a heterozygote that's intermediate, a heterozygote that exhibits codominance will show both alleles. Okay, so blood type is an example here. Okay, now blood type in the population, in the human population, there are 3 alleles. But an individual only has 2 of those alleles. Okay, so there's 3 out in the population of humans, but one individual only has 2 alleles. Okay, so remember on the blood typing, this is kind of a review for the final. Remember our red blood cells, if it's type A, it's going to have the A antigen or that A protein. If it's type B, it's going to have the B antigen. Maybe if it's B, well, B positive has this other antigen. If it's type O, it has none. And then if it's AB, do you remember it has both the A and the B antigens on those red blood cells? Okay, well, there's a genetic component that causes the different types of antigens on the red blood cells. Okay, so we'll take a look at this because this is a real example that you might be faced with in healthcare, is trying to determine a person's blood type. Okay. So the gene, well, let's go down here. Okay, so a person with type AB blood is going to show codominance. Okay, so both of the alleles are going to be expressed. You get a B and an A, so that's codominance there. Okay, that's where we get that. But let's look at the possible genotypes for each blood type. So a person with type A blood could have 2 dominant A alleles, or they could have a dominant A allele and a recessive allele. This one is recessive. A person with type B blood could have 2 B dominant alleles, or a B dominant allele and a recessive. A person with type AB, though, will only have an A dominant and a B dominant, and a type O will have only the recessive type. Okay, the recessive alleles. Okay, so let's go through this really quickly and I'll let you label. Okay, this one would be a heterozygous dominant. This is a homozygous dominant. This is a heterozygous dominant because it has a recessive allele. Homozygous dominant, heterozygous dominant, homozygous recessive, and this one is also heterozygous, but it has both dominant alleles. Okay, and this is where we see the codominance. Okay, so I have a blank slide in here, so you might want to grab your extra piece of paper, and we're going to go through a blood type example, maybe 2, we'll see. Okay, so mom, in this example, has type O, and dad is heterozygous type A. Okay, so what is the genotype for each of these parents? Look back on the last slide and take a look. Okay, so mom's going to be little I, the 2 recessive alleles. And dad is going to be big I with A. And since he's a heterozygote, little I. So we set up our Punnett square. little I for mom, big I, A for dad, and little I. So if we go across, we're going to have 50% chance type A as a heterozygote and a 50% chance for type O blood, which is going to be a homozygous recessive. Okay, let's look at one more example of this. Okay, here both mom and dad have type AB blood. Okay, so if we put up our Punnett square, mom is going to be A. Okay, so we'll fill this in. This one's going to be dominant type A. This one's going to be AB. This one's AB. And this one will be dominant type B. Okay, so they're going to have a 25% chance of being type A, which is this one. 25% chance of being type B, which is this one. And then a 50% chance of being type AB. Okay, so again, make sure you look over these and know how to determine a blood type based on the genetics. Okay, what alleles go for type A, B, O, and so forth, and how to do the Punnett squares. Okay, again, please let me know if you have any questions or you want me to review this with you. Okay, so there's some other factors that can affect expression of single genes. Okay, so sometimes genotypes vary a little bit from person to person based on the environment or the action of other genes. And that's when we get into some of these new terms here called penetrance, expressivity, and pleiotropy. Okay, so we're going to look at some of these variations in gene expression. And remember, gene expression is taking DNA, turning it to RNA through transcription, from RNA to protein through translation. So the term penetrance means there's basically all or none expression of a gene. So either a gene is expressed or it's not. Okay, and some genes fit into that category. Others are not. Okay, and expressivity determines the degree of a phenotype. Okay, the degree of which that gene is expressed. Okay, so complete penetrance is when a person inherits a disease-causing allele and they have that trait that expresses the genotype, or they have the trait, they have the disease. If you have the alleles needed, you get the disease. Incomplete penetrance is when sometimes some of the individuals don't express the phenotype. They don't develop that phenotype even though they have the alleles. An example of that is polydactyly. Does anybody know what that means? That's when you have extra fingers or toes. Usually here, there's an extra digit here after the pinky on the hand or the foot, okay? And so a lot of times the allele for polydactyly, since it's dominant, a lot of times people will inherit that allele, but they will not express that. So sometimes if the allele is present, some individuals will not express that allele, express that gene, and will not grow an extra finger. Sometimes people do. Okay, that's incomplete penetrance. Then another example of that is in an environment probably plays a role in that, okay, in determining which do and which do not develop that particular phenotype. Variable expressivity means that the phenotype might vary in intensity, and polydactyly is another example here. So if a person has that dominant allele, okay, one person may grow one extra toe or one extra pinky, and the next person may have 2 or 3 extra pinkies. Okay, and again, environment's going to play probably a role in that as well. Pleiotropy is the idea where a single gene, typically a single gene has an effect on a single trait. Okay, but pleiotropy is when a single gene produces several different traits. So one gene can cause different traits. Okay, so a lot of genetic diseases that would include this are when proteins or a protein is affected that would have effects throughout the body. And an example of a pleiotropic gene is the gene that caused Marfan syndrome, which is an autosomal dominant disorder. And the mutant gene causes a defect in a protein called filabrine, and that filabrine is going to be found in connective tissue, in elastic connective tissue, like in the aorta. Remember in the cardiovascular system, we said how the aorta expands and contracts and kind of pushes blood around to help maintain blood pressure. Okay, that defective filabrine would cause problems in the aorta and actually can cause a person to have an aortic aneurysm, which is where the aorta bursts, and death could occur very, very quickly because of blood loss. Okay, bones, ribs, the lens of the eye also has this protein. And so that one gene affects several different traits around the body. Okay, and so different people with Marfan syndrome have different manifestations of symptoms. Heterogeneity, though, is kind of the opposite of that, where you have one phenotype resulting from different genes. Okay, and that's typically occurs when different genes code for enzymes that work in different steps of a biochemical pathway. But there's a couple of examples here of heterogeneity. One is hereditary deafness. Okay, so this is genetic deafness. There are 200 forms of genetic deafness that are in which deafness is the phenotype, but different genes control different aspects of hearing, and so deafness can result from different forms of the genes. Also, blood clotting disorders, okay? With blood clotting disorders, some people might blood clot too little, okay, and they are at risk at bleeding, or people may develop blood clots that clot too readily and can cause stroke and things like that. But bleeding disorders can be caused from mutations in several different enzymes, which are proteins, that can result in the lack of the ability to clot. So there's several genes that could cause blood clotting if they have the mutant allele. Okay, so up until, oops, sorry, not there yet. Let's see. So we've mentioned before that genes can be, and gene expression can be affected by the environment. Okay, multifactorial or complex traits is when a gene or more than one gene combined with environmental factors cause a trait. So height and skin color are examples of this, as well as heart disease, diabetes mellitus, high blood pressure, and some cancers. Okay. Now, a lot of these are also what we call polygenic traits. Okay, which means that the trait is caused by more than one gene. Okay, so continuously varying traits are polygenic because there are many degrees of expression of those several different genes. Okay, so I'll tell you what this means here in just a second, but height, skin color, eye color, hair color, those are all polygenic traits. Okay, and so here is a continuously varying trait. Okay, so you have some really short people, kind of short people, a little taller, tall, 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 and really tall people. Okay, where you get most of them kind of around here in the median area. Okay, but it's continuously varying. You go from all the way really short all the way to really tall. Or in the case of skin color, okay, very, very pale to very, very dark. And you have variations in between. Okay, so these are polygenic traits. They're continuously varying and also influenced by environment. Okay, so if, for example, if this very tall person here did not have access to adequate nutrition, okay, and did not have protein and vitamins and minerals and things like that needed to achieve that height, he may be more down over here. Okay, so environment does play a role in some of this. Okay, there are 3 genes that control skin color, and each gene has 2 alleles. And so skin color involves, the model here shows 3, but there's probably more than 3 skin alleles, skin traits. But it's a continuously varying model here again. Okay, and it's the interactions, the expression of each of these 3 genes with their 2 alleles that determines which range a person might be in. Okay, same thing with eye color. Here is showing 2 different genes for eye color. Notice this Punnett square, it has 16 squares instead of 4 because we have 2 genes looking at here, the A gene and the B gene. They're probably representing the OCA2 gene and the HERC2 gene. Those control eye color. There's other genes as well that control the tone of the eye as well. Light blue eyes is completely recessive, like down here. Dark brown or almost black eyes are almost completely dominant with both of those alleles. Okay, so what we've talked about so far are the autosomes, okay, or chromosomes 1 through 22. Chromosome 23 does also affect our phenotypes, and there are certain disorders that are associated with that. So somatic cells are normal body cells, okay? The X and Y chromosomes, okay, are going to be those that determine the gender, and we've talked about that. So all eggs carry X's because a human or female can be XX, so all eggs will carry X's, and a Y can carry, a sperm can carry an X or a Y. So it is the male that determines the gender of the offspring, okay? Because offspring's gonna get an X from mom. If it gets an X from dad, it's female. If it gets a Y from dad, it's gonna be a male. Okay, so XX female, XY male. Okay, and this kind of just illustrates what I just went over. Eggs will always have an X. Sperm could have X or Y. X and Y combined to make male, X and X combined to make female. Now, embryos will develop as female unless there is a Y gene or a Y chromosome present. And so the male characteristics, or the offspring, will become male under the direction of this SRY gene found on the Y chromosome. Okay, this SRY gene has a called a transcription factor, which is really a protein that basically directs male structures to develop within the embryo and later the fetus, while suppressing female structures. Okay, so in the absence of this gene or this protein that the gene makes, the embryo and fetus will develop into a female if that's not present. Okay, now some traits, the X and the Y chromosomes have genes on them, just like all the other chromosomes. So some traits, some phenotypes are linked to the X chromosome, and some can be linked to the Y chromosome. And remember when we showed that karyotype, I mentioned that, oh, I can tell this one is female because it has 2 longer chromosomes, okay? The X chromosomes have over 1,500 genes, and they're very large. The Y chromosome is very short compared to the X. Okay, so you can see here, we have the X chromosome, and this kind of looks like an upside down Y. So basically what's happened is we have the X, here we have X, Y. This whole region of the Y chromosome is missing there. Okay, so it has many more, the X chromosome has many more genes on it than the Y. Okay, so in a disorder, like we'll talk here in a minute, like hemophilia or red-green color blindness that are associated with the X chromosome, here in the female, you have 2 of the alleles. In the male, you only have 1 allele because this one here is missing. And so whatever allele a male gets, if it's associated with the X chromosome, is what his phenotype is. Okay, and I'll go over that here in just a second. So what I've just described is that the male is hemizygous for the X-linked traits, meaning he only has 1 copy of the gene found on the X chromosome. So examples of X-linked traits are going to be red-green color blindness and hemophilia. Okay, no, those would be, yes, X-linked traits. Okay, so hemophilia is a blood clotting disorder, and where the patient would, if they got a small nick or cut, they would have problems stopping the bleeding. Okay, so this hemophilia can be passed on from a mom to a son, or could be to a daughter as well. The son has a 50-50 shot of receiving a recessive allele from a mom that's a heterozygote carrier. So a son that inherits 1 allele, 1 recessive allele, will have the disease. Okay, so say mom is a carrier, she has the allele there. Okay, the son will have a Y. If he happens to get this one that carries the allele, he will have hemophilia. If he gets this one here, he will not. So it's a 50-50 shot. Okay. Each daughter has a 50% chance of getting that allele from mom, okay, because she could get this one or this one, but she will have to get one from dad as well to have the disorder. Okay, so say the daughter gets this one. Okay, she's automatically a carrier, but unless dad has the disease, she will be the carrier. If dad does have the disease, okay, she gets the allele from mom and from dad, she will be affected with hemophilia. But it's usually pretty unusual for a daughter to get hemophilia because you would have to have a carrier or an affected mom and an infected father. Okay, so a female needs to have 2 of the alleles. A son would only need to get 1. Okay, I think in the interest of time, we're going to skip this part. Basically, there are things like hormones. that affect gene expression. So there are hormones that we saw help with breast development. There are hormones that affect some of these different traits here. And I wanted to finish up talking about chromosome disorders. So previously, up until this point, we were talking about alleles or single genes or groups of genes. Here we're going to look at the whole chromosome. Okay, so remember, humans have 46 chromosomes. And once in a while, it could occur that a person, a cell, a zygote, could get an abnormal number of chromosomes, and that would cause some issues. Okay, the term euploid or euploidy would mean having the normal number of chromosomes. Okay, so if a person has 46 chromosomes, they would be a euploid. A polyploid, or the condition of polyploidy, is having an extra set of chromosomes. Okay, and this would be very, very drastic. Okay, so this could happen in a case where perhaps you have, let's see, a sperm with 23, and for some reason the egg didn't separate. Okay, and you would get these two to combine together. This is what we call haploid, and this is what we call diploid. And if we put them together, 1 plus 2 would give us 3N or triploid, and you'd have 3 copies of each chromosome. And typically this would result in a miscarriage, but sometimes an infant can survive a day or two, but they're going to have a lot of abnormalities and will not survive long term. Okay. More often though, we see what's called aneuploidy, and that's when you have a missing A, or 1 missing chromosome, or 1 extra chromosome. And typically this occurs from what we call non-disjunction. And I'll show you the next diagram here and show you how what that means. But non-disjunction is basically when a chromosome fails to separate during meiosis. Okay, and so the zygote will end up with either 45 or 47 chromosomes instead of 46. Here, let's do the math, 69 chromosomes. Here, we're getting 1 extra or 1 less. Okay, let's see, trisomy is having the extra chromosome. Monosomy is when they miss a chromosome. Okay, and we're talking about autosomes here. However, the sex chromosomes can have an extra or a missing chromosome. And Turner syndrome is where you're missing, there's only 1 X. Okay, and the only detriment there is delayed sexual development. And this person would be female because there's no Y there. Klinefelter syndrome is XXY, and that person would be then male. Okay, if there's ever a Y, it's a male, and if there's only X's, that would be female. Okay, again, these are less severely affecting, and a lot of times these people will be able to reproduce. So here's the example of non-disjunction. Okay, so there's 2 rounds of meiosis. There's meiosis 1 and meiosis 2 to produce the oocyte and the sperm. Okay, so we have sperm production here. We could say the same exact thing for egg production. Okay, so normally in meiosis, we're going to have these chromosomes splitting out. So in a normal situation, you would have X here, this chromosome there. Okay, then this one will chromosome will go, or chromatid would go there and there. This is normal. You get one copy of this, one copy of that. Okay, however, in this example, these two stuck together. Okay, it's called non-disjunction. They don't pull apart. So this sperm got 2 copies of this chromosome. This sperm only got none of that particular chromosome. So if these two normal fertilize with a normal egg, here's the zygote. These guys are going to be euploids, which is normal. This one is going to be trisomic, has 3 copies of this chromosome, and this one only has 1 copy. These usually do not survive. Sometimes these can. If this is the 21st chromosome, this would result in Down syndrome, and this person could survive. A lot of times these monosomic ones do not. Another way that this could happen is if the non-disjunction occurred initially. Okay, you would have 2 sperm that resulted in a monosomic, and here you would get 2 with 3 copies. Okay, so I mentioned the 21st chromosome. Okay, if that one doesn't separate, that is Down syndrome. Okay, the chances of surviving that past 1 year is 85%, which is pretty good. Trisomy 18 and 13, okay, affecting the 13th chromosome or the 18th chromosome is more severe. Less than 5% survival rate. Okay, they're also more rare though. One other thing that I wanted to point out about Down syndrome though was the fact that chances of giving birth to a Down syndrome child increases with the age of the mother. Remember, she produces her oocytes before birth, and then they go on pause in meiosis. And so the longer meiosis is on pause, okay, the longer these divisions take, okay, or I'm sorry, the longer these divisions are on hold, the greater the chance of that non-disjunction, these two sticking together, or these two sticking together could occur. Okay, so that's why they say after 35, the chances of having a child going into or being Down syndrome is greater. It's because those eggs are on hold. Okay, now that's not saying that just because you're over 35 and have a child, that your child will be having Down syndrome. Okay, that's not the case. And I know a mother that had a Down syndrome child in her early 20s. Okay, so, and it's not always mom's fault. It could be the sperm that don't separate out, as we saw in our example. Okay. All right, just really quick to end up here. I know this one's kind of taken a little bit longer, but some tests that can be done. We talked about the Punnett squares and the pedigrees and things that can be done, but technology now has enabled us to look at the actual offspring conceived, and also we can do genetic testing on parents before they conceive. But this chart here tells us some prenatal tests, okay, some tests that we can do on the pregnancy to determine or to basically prepare if the baby is going to be born with some chromosomal problems. Okay, so an ultrasound can be done, and what they will do is they'll take some measurements of the head size and the crest of the neck and some other areas. There can be maternal serum markers, okay, where they would basically look at mom's blood and look at different components of that. There's chorionic villi sampling, which I'll show you here in just a minute, amniocentesis, and then cell-free DNA. This cell-free DNA is a newer technique, and it's starting to replace these two because there are risks with the CVS and the amniocentesis. A CVS is where they would go in with a tube and remove cells from the chorionic villi, or part of the placenta, okay? And here, amniocentesis is they would use an ultrasound guided needle to remove the amniotic fluid. And both of those procedures come with significant risks. This can actually rupture the bag, and then delivery would need to take place. And this can cause some issues as well. So the cell-free fetal DNA is a much safer method. Okay, here's a 13-week fetus that shows normal facial features. And the last two slides just talks about using genomics to kind of determine and predict different diseases. There's a big advancement in healthcare right now is looking at genetics and tailoring chemotherapies and other types of drug therapies to an individual based on their genetics and how they metabolize drugs and things like that to maybe be more effective. Okay, so don't forget to finish up the fetal pig lab before Friday and begin studying for exam 5 on Monday and your final exam either on Wednesday or Friday, depending on your which section you're in. Okay, again, please, if you need to go over anything or have any questions about the upcoming exams, let me know. But this is the last lecture that you guys will have for this class and this semester.